A warm welcome to all participants joining us from across India and around the globe. We, the students at People's University, are proud to present this special webinar driven entirely by students for students. On behalf of our organizing team at the Center for Scientific Research and Development, People's University, Bhopal, India, I warmly welcome you to the ninth session of the BioConnect webinar series, a unique for the students by the students initiative. My name is Aisha, and I am proud to tell you that I am a student of BSc Biotechnology and a member of this webinar's organizing committee. It is a great honor for me to serve as the MC for today's session. Today's webinar focuses on a timely and thought-provoking theme, biotechnology and precision medicine. We are deeply honored to welcome the eminent guests and leaders joining us today. A very special welcome to Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, our distinguished speaker, Professor Dr. Manikam Ravichandran, Scientific Research Professor Dr. Subhash J. Bore, our mentor and head of the Center for Scientific Research and Development at People's University, and Ms. Priti Kushwaha, chairperson of the organizing committee of this program. To formally inaugurate today's session, it is my honor to invite our Honorable Vice Chancellor, Professor Dr. Harish Rao, to deliver the opening remarks. Sir, please proceed.
Thank you, Aisha. Good afternoon, everyone. It gives me immense pleasure to welcome you all to the ninth session of the BioConnect webinar series organized under the banner of the Center for Scientific Research and Development at People's University, Bhopal. This series is particularly special as it truly reflects the BioConnect webinar series motto for the students, by the students. Today's event is yet another reflection of our students stepping forward with energy, initiative, and purpose. At the outset, I wish to congratulate the dedicated team of 6 biotechnology students who have taken the lead in organizing this event. Such student-led initiatives truly reflect the spirit of innovation, leadership, teamwork, pursuit for excellence that we strive to nurture at People's University. Today's theme, biotechnology in precision medicine, a collaborative approach to personalized healthcare, is timely and highly relevant. As we know, healthcare is moving rapidly toward personalization, where treatments are tailored to the unique genetic makeup and lifestyle of each individual. Biotechnology lies at the heart of this transformation, enabling breakthroughs that were unimaginable just a few decades ago. We are indeed privileged to have with us an eminent scientist and thought leader, Professor Dr. Manikam Ravichandran, Chief Scientific Officer at my home ALCS Global Holdings, Malaysia, an honorary professor at AIMST University, Malaysia. His vast experience in genomics and precision medicine will certainly provide valuable insights and inspire all of us, especially our students, to think boldly about the future of healthcare. This BioConnect webinar is not just about knowledge sharing, it is about collaboration, exposure, and empowering students to take ownership of their learning journey. I am confident that this session will spark curiosity, stimulate dialogue, and open new avenues of thought and research among the participants. On behalf of People's University, I extend my heartfelt appreciation to our distinguished speaker, Professor Dr. Ravi Chandran, for kindly accepting our invitation. I also acknowledge the guidance of Professor Dr. Subhash J. Bore, head of the Center for Scientific Research and Development, whose mentorship and vision has helped nurture this platform for academic and societal engagement. With these words, I once again welcome you all to this exciting session and wish the organizers and participants a very fruitful, enriching, and an immersive learning experience. Over to you, Aisha. Please take over.
Thank you, sir, for your motivating words. Your continued support to drive in students' academic initiative is deeply appreciated. Now I invite the chairperson of the organizing committee, Ms. Priti Kushwaha, to say a few words. Priti, over to you.
Asha, good afternoon, everyone. Our esteemed guest speaker, Professor Dr. Mani Ravichandran, our honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, our President, Professor Dr. Subhash Dore, respected faculty members, participants, and my dear fellow students. I extend a very warm welcome to all of you to this webinar session. My name is Priti Kushwaha, and I'm proud to tell you that I am an MSc student at Center for Scientific Research and Development, People's University. And I'm extremely honored to serve as the chairperson of this organizing committee for this program. On behalf of the organizing committee, I'm delighted to welcome you all to this thought-provoking webinar titled Biotechnology in Precision Medicine, a collaborative approach to personalized healthcare. This session will explore how advances in biotechnology are driving the development of precision medicine, which is moving beyond one size fits all treatment. It will highlight how collaborations among researchers, clinicians, and industry is making personalized healthcare more effective, accessible, and patient-centered. We, we are fortunate to have our distinguished guest speaker, Professor Dr. Ravi Chandran, to share his thoughts and insights with us on this important subject. With these words, I thank all once again for being with us today. Let us look forward to the exciting and thoughtful session ahead. Now I hand it over to our MC Ms. Asha. Over to you. Thank you.
Thank you so much, Ms. Priti, for your encouraging words and welcoming remarks. Fellow students and esteemed participants, it is now time to begin the core session. To take it further, I hand over the session to our moderator, Ms. Nandana Tiwari. Nandana, over to you.
Thank you, Ms. Aisha. Hello, everyone. A very warm welcome to all the participants joining us from India and around the world. I am Nandana Tiwari, an MSc Biotechnology student at the Center for Scientific Research and Development, People's University, Bhopal, India. It is my privilege to serve as your moderator for today's insightful session, organized under the BioConnect webinar series for the students by the students, a remarkable initiative of the Center for Scientific Research and Development, People's University, India. Today's session brings us to a highly relevant and timely topic in healthcare, biotechnology and precision medicine, a collaborative approach to personalized healthcare. It is my honor to introduce today's distinguished speaker, Professor Dr. Manikam Ravichandran. Professor Ravichandran obtained his MSc in Medical Microbiology from Christian Medical College, Vellore, and his PhD in Biotechnology from Anna University, Chennai. He has an outstanding research profile with 84 publications in international journals, and has guided 55 postgraduate students while examining 65 theses globally. His innovations include 18 patents, one of which sDNA-AMP diagnostic technology has been successfully commercialized into three diagnostic products. Professor Ravichandran has also served in several leadership roles. He is the former Chief Executive and Vice Chancellor of AIMST University, Malaysia, where he also chaired the COVID-19 task force. At present, he is the Chief Scientific Officer of ALPS Global Holding Malaysia, the first biotech company in the country to achieve unicorn status. His work has earned him more than 50 national and international awards, including the Idea Innovation New Product Award (IENA) in Nuremberg, Germany, the International Exhibition of Inventions Award in Geneva, and the National Innovation Award in Malaysia. In addition to research, he actively contributes to science communication as a columnist for the New Straits Times NST. Professor Ravichandran's career reflects a lifelong commitment to advancing biotechnology for the betterment of human health, particularly in the exciting field of precision and personalized medicine. With this brief introduction, it is my honor to invite Professor Manikam Ravichandran to deliver his talk on biotechnology in precision medicine, a collaborative approach to personalized Chandran. Over to you.
Hope you all can see my slides.
Yes, we can see. Please go ahead.
Okay. So thank you, Dr. Shubhas and respected Vice Chancellor, Dr. Harish Rao, and the whole team of students for organizing this excellent event. It's my pleasure to share um, a topic on precision medicine. So this I'll be talking on the application part of human genome sequencing. So it will be slightly different from what we learn in the classrooms. Mostly, since I'm in the industry, so I'll be talking about the most on the application part of the human genome. So if you see lately, this has started to stimulate you. The two prominent news recently you would have seen. One is about that Iceman, Udzi, and other one is about Turkana people in genome of Turkana people in Kenya. So Odzi is he is an Iceman. Uh, 3000 BC, actually, roughly around 5300 years ago. It's a well-preserved mummy in icy mountain. It's called Orzal mountain in Italy. So that mummy was found, and they took a biopsy from the ilium, and they have sequenced it fully, and it was discovered some time back, and now the whole genome is out. So actually, the genome has revealed what color of the eye he could have, and the hair and the skin tone, and also based on the genome of Otzi, that we know that he's lactose intolerance. He had cardiovascular diseases, and even had an infection of Borrelia burgdorferi. It's a spirochete disease. It's one of the oldest case of Lyme disease reported so far. And based on what is there in the tummy, they found that what meat he was eating a kind of a sheep meat, and what wheat and wild plants he had. He also had a tattoo, and possibly for the therapeutic purpose. It's just an example of an application of genome sequencing, how it was able to decipher demystify the history and ancestry and physical trait of mummy. In another example, recently in September 19th, it was published on genome sequencing on a group of people from Turkana, and they live in a harsh environment. They actually, the water is very less, but the diet has rich in protein and meat and blood and so on. So based on the genome sequencing, they have found that They have undergone a very strong natural selection, enabling the population to survive in the limited water supply, particularly with high meat and protein-rich diet. So, what was behind the genome was there is a gene called stenocalcin. It's a gene, it's a hormone gene, normally in the kidney. So it helps to conserve the water. So because if you eat a lot of protein rich, it has a lot of purine, it might damage your kidney. So it actually protected the kidney, and it it protected the people. But however, when these people migrate to the urban areas, so the urban lifestyle, these people might face risk of chronic diseases such as hypertension, obesity, kidney dysfunction, and diabetes. So this is one of the example how genome sequences help. So there are a lot of things we can talk about it in the current generation, how you can make a decision in your life. Possibly, you might be using the genome as one of the tool to see whether to move forward. So this is going to be just a preview of my talk. So precision medicine is a very broad area. It has so many other fields there, but I'll be focusing on the different types of genome sequencing and its applications and how some of the case reports and so on. I can do that. So precision medicine is basically is a tailored approach. Um, it's combination of lifestyle, environment, and genetic factors, genotype and phenotypic analysis. So it is utilized to make three things. One is prevention, diagnosis, and treatment. So precision medicine basically you can use it to detect a patient at the risk of enabling prevention strategies. Number two is to diagnose a disease based on the gene. Accurate disease diagnosis is possible, and I can tell you some stories about how they're able to diagnose some of the rare diseases using genome sequencing. And number three is about how we can give a targeted treatment based on the genomics. It's called a pharmacogenomics. So precision medicine is like customizing a treatment method for individual. It's like you go to a barber, you cut your hair. Each one has a different type of treatment when you go cut your hair. So similarly, in precision medicine, the amount of drug, type of drug, and all those things are customized. That's why it precisely treat and everyone get the benefit of it. Traditionally, what we do is normally, let's say a patient has a colon cancer, we give one drug. We don't know which target. Colon cancer could be different, maybe due to three different markers. So randomly we give a drug, maybe the drug will work only for one person. So other 2% it might not affect. So 1% it work. For example, if you use personalized precision medicine, if you use it, so if you know what type of marker the particular tumor has, based on that you use a customized drug, customized targeted therapy. So all of them will get benefited. So this is one of the example of the precision medicine. The precision medicine always started with the structure of the DNA, and Human Genome Project was started in the year 1990, and 2003 they completed, and it costed almost like 2.7 billion US dollar. But now you can do a human genome for less than $1,000. So this is how a lot of advancement has happened in the genome, and targeted therapy and pharmacogenomics are part of the milestone of the precision medicine, like a lot of targeted drug, checkpoint inhibitors, lot of things are coming. And pharmacogenomic also has come. You can treat a person based on the genes they have for drug metabolism. So poor metabolizer and fast metabolizer, you can prevent the drug toxicity, you can optimize the drug dose based on the pharmacogenomics part of it. So DNA technologies has been mind-boggling, and it's the technology You know that Sanger sequencing method and a lot of other methods you would have studied in the textbook. Now you have few generations of genome sequencing, like first generation, second generation Illumina, third generation of nanopore. Now it is nanopore is the world fastest. The Promethean 48 is the fastest human genome sequencing. You can sequence in a very short time, and when compared to the conventional DNA sequencing method. So there are many technologies involved in precision medicine. So sequencing is one, CRISPR, biomarker discovery, different omics and wearable technologies. These are part of the precision medicine, but this talk will focus only on the DNA sequencing. So you all know that every living organism has a cell, including human, and inside the cell you have the DNA. So inside the so basically in a human body have nucleated and non-nucleated cells. You have red blood cells, which almost is 29 trillion cells. Red blood cells. You have red blood cell does not have a DNA inside, but it 7 trillion of nucleated cells you have. So total of 36 trillion cells in your body, along with the 38 trillion bacteria. And so when these cells are there in your body, almost 1 million cells in the human body die every second. So you can say that in 80 to 100 days, uh, all the cells would have replenished, and then new you will be would have you said you should have a new cells inside your body. So almost like 330 billion cells are replaced daily. So that is the complexity of number of cells in the body. But if you go inside the cell, you have this 23 pairs of chromosome with 3 billion bases. So inside each cell, you have a 23 pair of chromosome, and it's happening in every 30 trillion cells, the nucleated cells alone. And it has this DNA basis. You all know that as a biotech student, you know that it has ATGC and it's also located. And these chromosomes are tightly packed with the DNA structures. So genes are expressed only when it is needed based on the promoter and operator and so on. So the genes coded by the triplet, you all know that, and it is transcribed. So the central dogma of molecular biology, the process of gene expression, um, this is very, very important. This is one of the cardinal point, the dogma you need to understand. So from the DNA, this is a primary transcript is formed, process called transcription, and you have a mature RNA which removes the introns, which removal of intron and splicing happens. Then your translation called protein. The whole process is called central dogma, and this is the process of gene expressions. So if you look at the human genome, this 3 billion bases, so you have this promoter, intron, and exons. Introns are intervening sequences. They do not code for any region, but it has some regulatory function in rare occasions. Exons are the actual protein coding sequences. So this is what we study in our classroom, okay, central dogma, how we translate into DNA sequencing. So there are many terminologies we used in whole genome sequencing. It's called, first one is called whole exome sequencing, WES. So basically whole exome sequencing sequences all exons, the genes in the human genome. We have around 22,000 genes. It represents only 1 to 2% of the 3 billion bases we have in our each cell. So the WES is called whole exome sequencing. Whole genome sequencing sequence the whole thing, including the intron and exons, everything, 3 billion bases are sequenced. That is called whole genome sequencing. Then you have a transcriptome. You targeted sequencing. If you know the mRNA sequence, design a primary probe, and you can target and sequence, that is transcriptome. And epigenetics basically look for the methylation site. There is a special method to protect the methylated DNA and then sequence it and identify epigenetics modification happened on the genome. And there is another method which is currently used for cancer diagnosis is called circulating DNA. So it can be any form, either genomic DNA or mRNA or any of the fragments of the DNA of roughly less than 200 base pair in size that it circulates in the blood. So they are detected and for cancer diagnosis. So basically what happens is, though there are 3 billion bases in the genome, there are some mutations. You know what is a mutation? There's a change in the DNA base, and normally the DNA sequence will have a reference SNP ID cluster. You know, SNP is a single nucleotide polymorphism, which is there in our genome. Each has an ID clusters. So when the genome sequencing is done, you normally will get a VCF file, variant call file, where all the mutations will be piled together in this variant file format with the ID, what type of mutation, insertion, and reference sequences, everything will be there in that file that is uh, used to um, used to extract the information on the mutation present in different genes in the genome. So different genetic marker present in the genome, as you can have a risk of cancer, diseases, wellness and fitness condition, and drug response, and so on. It can be easily identified by this genome sequencing. So coming to the mutation, you have to understand two type of mutation in a human body. One is a germline mutation. Germline mutation means you inherited from your parents. It's called germline. So from the germline, it is there in all the uh, 3 billion bases of your genome. So it is basically passed from parent to child, inherited, exists in every cells in the body. And so this is called germline mutation. Somatic mutations are acquired mutation. These mutations are due to cancer. Okay, there's new mutation happen at one particular position. It is not there in other cells. It is in one particular position. So that is acquired person's lifetime in a single cell, but it keep multiplying and it transfers. So since it's not a germline, this mutation might not be. It will not be inherited to the next generation. This is in a different location and a separated site in the body. So these are the normal terminologies used. One is called germline mutation, another one is the somatic mutation. Somatic mutation mostly due to cancer, you have this mutation, germline mutation, which we inherited from the parents. So there is the different type of sequencing which I mentioned before, WES, WGS, targeted sequencing, circulating tumor DNA, and epigenetics. So first we start with whole exome sequencing. Whole exome sequencing is a genome test which sequences protein coding regions. Okay, more than 22,000 genes is present in the human genome, which will be sequenced by this area. It's only the limited coverage, which we will not be sequencing the exon region and repetitive regions are not sequenced. One of the advantages is fast, it's inexpensive. Only 2% of the 1 to 2% of the genome we are sequencing it, and it's cost effective to do that. So we sequence only the exon region of the particular genome, and it is targetedly the sequence is the DNA sequencing is done, and you will get the output from the WS whole exome sequence. For the whole genome sequencing, so we take the whole genome, including the intron, exon, and other regulatory element, all the 23 paired chromosomes. 100% of the genome will be sequenced. It's called whole genome sequencing. And one other thing we need to see is the coverage, how many times we cover, because when you do the genome sequencing, you always ask for certain things. One is the coverage. So what coverage it is means, if it is 100x coverage, means it is sequences done in a very comprehensive manner. The coverage and the accuracy of the data will be very good. So this is the whole process of whole genome sequencing. Normally, you can start from a saliva sample or buccal swab or from a human blood sample. Human blood sample normally yields very good quality DNA, and this DNA is shared. Depends on the type of sequencer, whether it's a nanopore or NGS, Illumina-based sequencer. So you have to use the particular kit and processes and libraries created and sequencing is done. So basically, WES whole exome sequencing is around 60 million bases, but whole genome sequencing is you get a whole 3 billion bases. So the key purpose of WGS is help to predict the chance of developing hereditary cancer, and any changes happening in the genome data all can be identified by WGS. So the third method is called targeted gene sequencing. Targeted gene sequencing is basically they use a method like a PCR or microarray. They are cost effective. So you target certain gene. Let's say you want to sequence P53, BRCA1, CRAS1, BRAF1. So you know what genes you want to sequence, and you just amplify that, reverse transcribe the mRNA, and then sequence them. That is the targeted gene sequencing. So we take the MHA mRNA transcript, convert into cDNA, and then sequence those. It's very simple to do, and if you give a if you know the gene, you can just sequence it by this transcriptome. The technology they use normally by polymerase chain reactions or microarray methodology. So at my genome in our company, we offer 605 gene panel, which can predict up to 15 diseases. All these conditions can be diagnosed. And those particular gene probes are there in the panel, and it also has a cancer panel, and we can detect it. Normally, the reports, you as a student, you have to remember the reporting. You cannot talk science to a clinician, molecular biology, and you have to be in a very presentable format. It's like a heat map or like a traffic light, green, yellow, and red. So red means it's a mutation or things like that. So you have to present the report in a presentable format. So that's how it is reported. ctDNA is also called the circulating tumor DNA. It's normally when there is a cancer, the cells will be dying and going around in the bloodstream. So they also shed some of the DNA into the bloodstream, so it can be easily captured and sequence them. That is the circulating tumor detection. There are many methodology to do it. There are many different methods. Basically, the size of the DNA we detect by circulating DNA detection is around 200 base pair. So normally they take the blood and they separate the cells and extract the DNA and then detect by DNA sequencing, or they also do immunofluorescence or immunohistochemistry or single cell sequencing or PCR also been done by this circulating tumor DNA detection. The last method is epigenetics. Epigenetics, basically, there is no change in the DNA sequence, but there are some methylation on the DNA sequence. So there is no alteration in the DNA sequence itself, but we study. There are methodology which can protect the methyl group, coated DNA, and we can sequence them, and that has been associated with certain disease conditions. So methylation can be detected by the epigenetics detection kits, and you can identify the methylation in DNA sequence. Uh, Methylation can be two major types of epigenetic markers. One is the direct modification of the DNA strand by methyl chemical group called methylation. And sometimes they also histone modification also can happen. So when there is methylation, the two things can happen. One is silencing gene expression or enhancing the gene expression. So this can either silencing or overexpressed of the gene can happen. So hormonal dysregulation in the adipose tissue, Several type of cancer are under epigenetic control. SIND1 gene binding to the methylated arginine promote angiogenesis in human hepatocellular carcinoma. So there are so many markers already there. So based on that, we can identify the epigenetic markers on certain gene and diagnose certain diseases. So how whole genome sequencing is useful? They found that in colorectal cancer, CRC, there are some deletion and some fragile sites they identified as being associated with the treatment efficiency. Based on the mutation, you can say how the treatment will respond to it. So so these are the different method. I think we saw WES, whole exome sequencing. I know, I think as a student, you know what is exome. The whole genome sequencing is genome sequence, whole genome, and targeted sequence by transcriptomics and epigenetics and circulating cancer DNA detection. But so these are the different types of method we have discussed so far. So there is another area. It's called pharmacogenomics. Pharmacogenomics also is from a personalized drug therapy. So it tells you based on certain genetic makeup how the drugs are metabolized in your body, and how the treatment can be developed. This is an example. This is a simple example. It may not be correct. So let's say for someone with a different genetic makeup, only two Panadol will work. Paracetamol will work. For some of them, even half of the drug will work. So it based on the genetic makeup, the drug metabolism. The one who require the higher dose, may they're the fast metabolizer. The one who require the less dose, possibly a poor metabolizer of the drug. So based on the gene, example is cytochrome. P450 genes are involved in the drug metabolism. Based on the different form of this cytochrome gene, so you can identify how they metabolize the drug and so on. Example of pharmacogenomics in oncology, EGFR mutation. If the patient has a EGFR tyrosine kinase inhibitor, this particular drug can be used and so on. So based on the mutation, certain drug can be modified and used. So when we generate in our company, when we generate a pharmacogenomics report, we classify into three: strong, moderate, and weak. Level 1, level 2, and level 3. Level 1 means there is a high, because genomics is a developing area. A lot of studies been done, evidence has been published, experiments has been done. So based on the evidence and supporting publications, everything is there. We say level 1A. Okay, so it's a highly predictive. So sometimes there is no evidence, means it can be level 3. So based on that we report. So not all genes and not all drug, the data is available in the literature. Based on whatever is available, we can give. This is an example of one of the pharmacogenomic report. For example, we can say that for cancer, which drug will work, for epilepsy, asthma, and so on. I give you some of the few examples here. For example, pain. Somebody, this is one of the patients' report. This is the SNP number, RSID, and this particular patient with this genotype may require an increased dose of fentanyl for pain. Okay. Example for neutropenia, this particular patient is increased risk of neutropenia if you want to use irinotecan. So you have to be very cautious when you use it or try to use it. So toxicity is there. So sometimes the advice could be a dosage, advice could be a toxicity. Toxicity, just try to move out to a different drug. Don't use this drug. Okay, this is based on the evidence. So this is the evidence is 2A, is moderate. This example is also 2A. So example, salmeterol, patient with this asthma may this particular patient with this genotype, A genotype, may have a decreased response to salmeterol. So based on the genome, you need to alter the dose, and you can advise or go for a alternative drug for this particular patient. So this is some of the example of the pharmacogenomics report. Whole genome sequencing also, the reporting is very important. So when you report to a clinician this particular thing, you cannot talk about mutation and so on. You just say we normally say high risk, normal, or sometimes this mutation give advantage to the patient. Okay, we can say high risk for allergy, but for this particular patient is the 8 high risk to normal to advantages. So what are the high risk? Allergic to dust mites, egg, milk, and so on. Normal. What are the genotypes are normal? What are the what are the allergens which this particular patient have resistant to this kind of allergy, like a grass allergy and mugwort allergy and so on. So his example, so it gives a detailed report why high risk for allergy to egg, and what are the SNPs, the mutations respect to that. You can, if you want to study more about it, you can search this in the SNP data, single nucleotide polymorphism database. You can learn more about it and advise the patient. The most important aspect of the report of human genome sequencing is how it is interpreted, how it is presented to the patient is very important. So every company should have a genetic counselor. Normally should have a capability to read the genome, understand the genome, explain the findings of the genome to the patients. So because it's that if a gene is there, it's not exactly, it might going to cause that particular thing. Sometime the environmental factor, the epigenetic factor might overrule the function of the gene. So you have to understand the particular report with caution. So this is an example of allergy to the mold. This is the SNP sequences and so on. This cardiovascular wellness, particularly this particular patient is cardiovascular diseases. If you go to the particular page, it tells you what mutations that particular patient has, and it has all those stuff there. And coronary heart disease, these are the mutations this particular patient has. So this is some of the example of from database from a website called Undiagnosed Disease Network, UDN. So they found correct diagnosis for this particular patients, and they identified the polymicroglia linked to a GPR56 gene based on the whole genome sequencing, and they were able to avoid a brain surgery and improve a diagnosis. This is an example of whole genome sequencing from a person with epilepsy, okay, in fact, with the refractory seizures. So they did a genome sequencing. They found this particular anticoagulant deficiency. So it is due to the deficiency of the vitamin B6 pyridoxine. So they started the vitamin, and they have a dramatic improvement. If they have not diagnosed it, they would have treated this patient with anti-epileptic drugs. So it helped to diagnose the disease in a very precise, uh, very accurately. The disease was diagnosed. So this is also another example using ketogenic diet. There was a significant improvement in the management of the patient. This is a very interesting story of twins, and the two boys. They had something called Niemann syndrome, and they had a cerebral palsy, and you know that was the general diagnosis. But when they looked at the genome, they found there was a faulty gene in the mitochondrial fatty acid synthesis. So just by giving supplement of lipoic acid. Basically, this gene involved in mitochondrial fatty acids. This pathway, there was lipids. Was blocked. So when they supplemented with very inexpensive this supplement, the child milestone and everything is improved. So all the milestones and they started improving it. So this is one of the another good example how whole genome sequencing helped to diagnose the disease accurately and treat the patients. So this is the website of Undiagnosed Disease Network, and they help to do that. So we are not too far away. I won't say future. The current scenario of patients and doctor, you might even have a situation that doctor, can you check my genome and diagnose my disease? So this can happen very soon. So now the genome sequencing is affordable, and the quality is very important. So when you want to sequence, you need to always ask for whether the lab is ISO accredited and the quality of the DNA sequencing, whether it's a 10 times coverage or 100 times coverage. 100 times or higher the coverage, the more accurate your genome data would be. So all those things you need to follow. The most important is the privacy. You send your DNA overseas and the data get leaked. So you have to make sure that the data protection is of your genome data is properly protected, and all the privacy is done during the process of genome sequencing. So the AI also will play a role in helping to identify the human genome. So I asked the question in the beginning, can I have a look at the genome before I make a decision? Yes, this can happen. In future, the younger generation might need to look at the genome, not only infectious diseases, you need to look at the genome before you make a decision in your life. Genome compatibility profile, I was just searching. It's also there. There are certain apps and website that does this matching based on the genome. So in summary, I think precision medicine is a very growing area. It has a great application in our health, lifestyle improvement, and many other factors. And there are many type of genome sequencing. You have exome, genome, targeted gene sequencing, and other different method of DNA sequencing are there. So this is what we talked about it. And ALPS is the company where I'm in. We have medical center, wellness center. My genome is ISO 13485 accredited facility. We also have a stem cell facility in our company called it's a CGMP facility, and it's located in Kuala Lumpur, Malaysia. So I'll be glad to answer. I will stop my slide, and if any questions, I'll be very happy to answer anyone. Thank you.
Thank you so much, Professor Ravichandran, for that insightful and inspiring presentation. You have given us a deeper understanding of how biotechnology is shaping the future of healthcare by making it more precise, personalized, and collaborative. We will now move into the interactive Q&A session. Participants, please post your questions in the chat box and use the raise hand option if you would like to speak. While we wait for your questions, I will begin with a few questions to get us started. Professor Ravichandran, precision medicine often requires integrating genetics, clinical, and lifestyle data. What are the biggest challenges in achieving this integration, especially in diverse population?
Yeah, there are a lot of challenges in genome sequencing. You know that there's so many different platforms are there. You know about Illumina sequencing, and now you have a nanopore sequencing. The data format is slightly different, so you need to make sure that the data are analyzed in a proper format. Number two is the particular precision medicine. There are so many inputs from different formats. So let's say you have wearables, and they have different. Now you have a continuous glucose monitoring. So many other wearables are there. The data formats are different, and it has to be harmonized. Number two, how many clinicians know this kind of knowledge? And how many scientists know this knowledge? So there's a lot of huge volume of opportunities are there in this. I would take it as an opportunity. So you need genetic counselors. So these are the challenges. Finding a good right genetic counselor with the knowledge in genome sequencing, with the knowledge in nanopore technologies, is very, very rare. As a biotechnology student, you can take up all this stuff and then learn a little bit of everything. So nowadays, it's a challenge is you have to be a multidisciplinary or transdisciplinary. So all this integration is very important. Most important is the data privacy. Okay, so data privacy is very important. There is an online course by WHO. You have to can try taking that course. It will also help you. So data privacy is very, very important. Integration with the clinician, clinical knowledge, educating the clinician to apply this knowledge. We might do all kind of wonders in DNA sequencing if we are not able to teach the clinicians on the right way to interpret the data, and it is always challenge. So these are some of the challenges we face particularly. And as you know, the genome is not like a hard and fast rule. Some of the gene might be overruled by epigenetics. Um, but there is a risk there. You have a mutation, means there's a risk there, but it may or may not happen. So it's very, very, you have to explain to the patient, explain to the doctor in a more realistic way. Uh, some of the patient, oh, you have a mutation in cancer, you're going to die, and the patient will die not due to cancer because he got scared of the mutation. So you have to be very, very cautious because you have 3 billion bases. You might have millions of mutations in a genome. By looking at it, we don't want to scare the patients. So you have to tell them, no, you have a risk there, but you have to take precaution. You have a risk for cardiovascular diseases, take precaution. So you have to use your knowledge very carefully, particularly when you apply this knowledge in the clinical situation.
Understanding these integration challenges sets the stage for our next question. So, my next question is, what, in your experience, how is biotechnology transforming the affordability and accessibility of precision medicine for developing countries?
Yeah, I think I mentioned in my slide, when they start the Human Genome Project, the cost of the human genome sequencing was how much? It's around 2.7 billion US dollar. Now, the cost, less than 1,000 US dollar. I think there are companies challenging now. You can even get $100 genome. So the affordability is not a problem. It has beaten the Moore's law. The computer processing speed capacity is increasing and increasing, but this one will beaten up everything. The cost of actually extracting the DNA, sending the DNA will maybe more expensive than the actual DNA sequencing. So you have Prometheon. It's one of the fastest DNA sequencer now available. I don't know how you've seen nanopore sequencer. It's like a small device you can attach to your laptop and you can run it. So Promethean is quite big, but if you see a nanopore DNA sequencer, it's very, very small, like a disposable sequencer. So it is you can do a DNA sequencing at home if you have all the DNA extraction and this nanopore devices. So affordability is not at all a problem, and so this is Accessibility is also very important, and is actually, though it is affordable, it is the whole process is a multidisciplinary. You have a technician should extract good DNA, and you need a person to run the DNA sequencer, and you need a bioinformatician to interpret. Now you can use AI, but it's still emerging now. Emerging, the technology is emerging. I think you can put your effort in learning AI, all those stuff to understand. Bring the analysis part. So data is cheap, but analysis you need real human to analyze it. That's the most expensive part now. So affordability is possible, but we need a lot of brain power to interpret the bioinformatics knowledge to translate in at the clinical point of view.
Affordability is crucial, but it cannot be achieved by one sector alone. So, could you share your views on what role does collaboration between academia, industry, and healthcare institutions play in advancing personalized healthcare?
Yeah, the collaboration is very important because I'm a scientist. I worked in the university for 25 years. Okay, I did a lot of research. I developed vaccine and everything. You can do up to the developmental stage in the university. Okay, if you want to translate the technology to the clinical stage, you need to work with big pharma companies, and they are listed, and they have investors to invest on the technology or the patent, and it can develop into, it can go for a preclinical trial, phase one, phase two, phase three clinical trial. So it's very imperative to collaborate with the healthcare industries, and they will help you in the translational pipeline, and so that it can reach, the technology can reach the people. So and also the regulatory requirement is also very important. So you need to, whatever you do, keep the end in mind. At the universities, we do experiments, but most of the data they don't accept. It has to be done in a GLP, good laboratory practice. Whatever the clinical trial you do, you should have a good clinical practice. Whatever documentation you do, GDP, good documentation practice. So all those things you need to adopt it to bring a collaboration in a fruitful manner. So that is the hope I answer your question.
Collaboration
And then,
often leads to real breakthroughs. Speaking of which,
yeah.
let's turn to some of the recent advances in genomics. Could you share your could you share some recent breakthroughs at My Genome or in the field of genomics that you find particularly promising or personalized treatment?
Yeah, I won't say it's a breakthrough. It's bringing a new pipeline is always challenging. Okay, when you do the whole sequence, the whole genome, we have so much of data there. Okay, we work with neurologists. They give input on genes associated with the neurologists. We generate a neurology report because you have neurology is a broad field. So we developed a disease for Alzheimer. We developed a report for Alzheimer disease, Parkinson disease, autism, and things like that. So when you collaborate with people, maybe if I work with a nutritionist, I might come up with a report on obesity. So you can come up with different type of formats of report when you collaborate. So we have come up with the pharmacogenomics report that is actually very rare to see. So based on the drug metabolism, what are the genes there, and we have come up with the pharmacogenomics reporting from the whole genome sequencing. So whole genomic sequencing price is there, but when we generate the report, the cost will increase. So this is how we developed different type of reports, and also at My Genome, we also started developing our QC laboratory. Any methodology, you need a QC. So this is particularly for cell therapy. We have our own QC service, like looking for endotoxin, not for DNA sequencing. This is the cell therapy, a stem cell. So we have a lab that can do this kind of stuff. And we also keep watch on new technologies like CRISPR, gene editing, all the new methodologies. We work towards that. And also we have three AI engines now. So now we not only work in the wet lab, so we have our own, I forgot the name. There is NVIDIA processor. We have three server. We are now teaching the AI like a mini chat GPT. We call AlpSeek. We are developing our own AI engine, which can not only use as an AI asking general question, but it can decipher the DNA, human genome, DNA, all the analysis which can be done in a faster manner. So we are trying to have a subsidiary company that can use the AI to predict the genes and annotate the genes and so on. These are some of the things we are working on it.
Breakthroughs are
Yes, Nandana.
exciting, but they also raise important ethical questions, especially around data privacy and genetic testing. So, my next question is ethical concerns around data privacy and genetic testing are often raised. How do you think these issues should be addressed to build trust in precision medicine?
Yeah, as I tell you, ethics are very important. Okay, particularly in precision medicine, and you have to remove the data. You cannot talk about the name. You have to remove the ethnic of the person. All has to be removed, and it must be a part of the curriculum now. If you ask Dr. Subhash, I would say that the data privacy is very, very important. So it has to be taught, and there is a WHO standard available. You can do a WHO course, and then you have to protect the data privacy, because if the data leak to an insurance company, you're gone. You never get a policy on the medical insurance policy. And people can target based on the susceptibility of an individual to certain genes. So your most important thing, when I want to send my DNA sequence, I will ask for. I don't send to a faraway place and they will sell my data. No, I send to a secure place. They have their own server. At my genome, we have our own server. We don't send the sample outside. The data privacy is tightly protected, and you make sure it is not hackable. So the server all should be protected, and you should have a clear consent from the patients, parents, and it should be transparent. The literacy is very important. You cannot WhatsApp things to people like that. You have to be very careful. So data privacy is a very important element in human genome sequencing service.
While ethics ensure public trust, the future also depends on the next generation of scientists. With that in mind, what skills and knowledge should today's biotechnology students focus on to contribute effectively to precision medicine in the future?
So first of all, you have to be a good human being. Okay, that's very important. You follow the ethics. These are two important things which is lacking in the society. Everyone is after money. You follow your own ethics. Okay, that's very important. And next comes the knowledge. What knowledge you need to know? I told you right how to interpret the data. We need people to understand the genomic data. Now the AI is coming in, and you learn AI, like learn some of the basic scripting language like Python, Perl, and R. There are few basic scripting languages you can learn, and learn machine learning, and learn prompt engineering, and all those modules you can learn. It's actually it's not a rocket science. It's very, very easy to learn, and but don't totally depend on AI. Uh, so this recently one of my intern came and asked me, Prof, how did you design vaccine before AI? We used to think beyond AI those days. So that's why you're able to do now. AI can answer any questions for you, but you need to have some strong foundation to understand. So bioinformatics is a good area. You can learn. It's like a hobby, you just go online and learn how to annotate a genome, how to understand the genome, what are the methods available, how to give genetic counseling. Maybe you can do. You need to learn. You need to do courses like GCP, good clinical practice, good documentation practice. That will give you a highly paid job in pharmaceutical industries. Okay, so to learn all those extra stuff, don't just come out with a MSc or BSc with biotechnology or whatever degree, but learn additional courses like this, which will enhance your market value and be a good human being. That's more important.
Students will play a key role, but they won't work alone. They will have a powerful tools at their disposal. Looking ahead, how do you envision the role of artificial intelligence and machine learning in accelerating discoveries and applications in precision medicine?
You cannot run away from AI. You must learn AI. All those LLM or whatever law modules you need to learn, and don't totally depend on that. But give I'll tell you one small example. Uh, when you want to develop this kind of AI, just teach them properly. Also, when you ask questions, also you ask in a more technical way, then you get the correct answer for that. So most of the AI, they won't ask you back. They just immediately give you some answer. The answer may not be correct. So you have to learn AI. Always verify the answers, whatever the thing gives, and your your knowledge base. Normally they call in when we develop AI, it's called knowledge base has to be robust and strong. But the internet has a lot of wrong information. Okay, particularly in publications, you have a predatory publications, you have a you pay, and now you have AI generated publications also available in the net. So you have to really filter the right knowledge to the AI so that you get the right output. So you have to integrate AI in all your work. It can definitely save time, and so that results are usable.
Thank you for those thoughtful insights. With that, we have come full circle. From today's challenges to tomorrow's opportunities, gained a rich perspective on the future of precision medicine. Hello, fellow students and esteemed participants. Do you have any question? Please unmute yourself and ask questions.
Okay, we can ask questions. Don't get scared. And if you want any questions in any part of it, I think Dr. Subhash, you want to ask something?
Yeah, yeah. Professor Ravi, thank you very much for very informative talk. So, in audience, we have MSc student, BSc student, and biotechnology student. So maybe you can share your, words of wisdom for their future career, how they can think of future making career in genomics or in biotechnology.
There are a lot of opportunities in genomics, I can tell you. And there are a lot of, yeah, yeah, a lot of opportunities. Let me answer, Dr. Subhash. So there are a lot of opportunities. A lot of opportunities are there in in particularly the bioinformatics area, and you can learn by yourself, and you can develop a lot of apps, and you get involved, and you can move forward. But whatever you're doing, BSc, MSc in biotechnology alone, you try to do certain things which add value to your course. That's very important. The GXPs, all those GCP, GLP, all those courses you can do. And you can also diversify into bioinformatics, learn some of the basics, and you can also become a genetic counselor if there is any certificate course in genetic counseling or something. Just do that, understand it. A lot of people will be looking for people like you to read and understand genome, this genomic results. So the next is Prabhat Kumar. You want to ask something?
I can see the raised hand of Prabhat Kumar. Please unmute yourself and go ahead.
Dr. Prabhat, you can unmute yourself and you can ask question. Dr. Prabhat. Maybe by mistake he raised the hand.
Yeah, it's okay. So I think everyone is very serious
Sir,
in
I wanted to ask
this.
a question.
Okay. Yes, Wasika. Wansika.
Sir, what is the role of biotechnology in precision in medicine?
Yeah, biotechnology plays a major role. Precision medicine has so many components, your wearable devices, and many methodologies are like gene therapy. Many things are there. And if you take biotechnology, I would say two arms. One is the sequencing part, all the whole genome, whole exome, transcriptome, all those sequencing part comes in. And the gene therapy is also part of precision medicine. You can it spans between these two areas. Gene therapy is a uh, very potential area which is underdeveloped, and it is doable. Changing base is not that difficult nowadays. So you can use adenovirus or lentivirus to deliver the genes, and so biotechnology, I would say it will take the major chunk of the precision medicine. It has a lot of opportunities. CRISPR, gene therapy, bioinformatics, genome sequencing, these are all a part of the precision medicine.
Thank you for addressing our questions so patiently and thoughtfully. We wish we could have more time, but we don't. Dear fellow students and esteemed participants, that bring us to close of a truly enriching session. I take this opportunity to thank Professor Dr. Manikam Ravichandran for sharing his thoughts with us today. A big thank you as well to all the participants for your engagement and thoughtful questions. With that, I now hand the session back to our MC. Ms. Aisha, over to you. Thank you.
Thank you, Nanda, for effectively moderating the session. Distinguished guests and fellow students, as we draw this exciting session to a close, I now invite Ms. Celine to deliver a vote of thanks on behalf of the organizing committee. Celine, over to you.
Thank you, Aisha. Good afternoon, everyone. It is indeed an honor and a privilege to deliver the vote of thanks on behalf of the organizing team and the Center for Scientific Research and Development, People's University, Bhopal, India. As we draw the curtain on today's enriching session of the BioConnect webinar series, we are truly grateful to our distinguished speaker, Professor Dr. Manikam Ravichandran, for his valuable time and insightful talk on biotechnology in precision medicine, a collaborative approach to personalized healthcare. Sir, your thoughts have enlightened us and provided meaningful insights on biotechnology in precision medicine. We express our deep gratitude to our Honorable Vice Chancellor, Professor Dr. Harish Rao, for his esteemed presence. Your encouragement and continuous support serve as a great source of inspiration for all of us. We extend our sincere gratitude to our mentor and guide, Professor Dr. Subhash J. Bohre, head of the Center for Scientific Research and Development. Sir, your vision, guidance, and constant support have been the driving force behind this webinar series. Your ensuring encouragement has been the backbone of today's successful gathering. Heartfelt thanks to our moderator, Ms. Nandana Tiwari, for conducting the session with clarity, confidence, and depth. Your thoughtful questions brought out the best insights from our speaker. We are thankful to our chairperson, Ms. Preeti Kushwaha, for her support. Her efforts are appreciated. Before we close, I would like to thank each one of you present here today. The active participation of the audience with thoughtful questions and attentive listening made this event truly engaging. Knowledge grows best when shared, and today's session has been a shining example of collaborative learning. We hope the insights gained will inspire further exploration and innovation in the field of precision medicine. Finally, we want to record our special thanks and most profound appreciation to the management of People's University for their unwavering support and encouragement. Support of management for student led activities such as the BioConnect webinar series enables us to dream big and achieve more. With that, I hand it over to our MC to continue with the proceedings. Miss Aisha, over to you. Thank you.
Thank you, Salim. Once again, thank you to everyone who joined us today, especially our speaker, Vice Chancellor, mentor, and participants. It was a great session today. Before ending this session, we would like to hear feedback from all participants. The post-webinar link is posted in the chat box. We promise the evolution survey will not take you more than 2 minutes. Your feedback will be greatly appreciated. We will wait for a few minutes to let the participant complete the feedback form. We hope you have completed the feedback form. Please note that your e-certificates will be sent to your registered email IDs shortly. We sincerely thank you once again for being part of this enriching session. We look forward to welcoming you to our next BioConnect webinar very soon. Until then, stay curious, stay inspired, and keep connecting. This is Aisha signing off on behalf of the CSRD students team at People's University. Have a wonderful evening.